Hello everyone, I am Kamalesh Damodaran, founder of ScientiFlow. Today I will show you how to perform protein-ligand molecular docking using ScientiFlow. Let's get started. Docking workflow in ScientiFlow is very powerful. It will allow you to perform varied range of docking. First of all, docking can be broadly classified into two types. One is blind docking, another is site-specific docking. With each of these types of docking, one can further do three methods of docking. First, forward docking, and reverse docking, and then cross docking. In forward docking, one protein will be docked against multiple ligands. In reverse docking, multiple protein will be docked against one ligand. In cross docking, multiple protein will be docked against multiple ligands. For each of these docking methods, you will have choice to select varied range of scoring algorithms like Vina, Vinardo, AutoDock 4, AI-enabled scoring methods, and more. All this can be performed through our single workflow. Now, let us prepare the sample to perform docking with ScientiFlow. You can prepare sample with any of your favorite protein visualization tool like Chimera or Pymal or as you wish. I am using Chimera. This is example protein structure I downloaded from RCSB PDB. You might notice that more than one chain of the same protein is present there. I will be wanting to keep only one protein and then do the docking. To do this, I am simply selecting the chain I do not want, and then I am going to take actions, atom, delete. Now I am just keeping only the chain A which I am wanting to perform docking. Now next step is remove all the water molecules and then ligands and unnecessary metal ions. To do this, I just go to residues and then select all non-standard amino acids, go to action, atoms, click delete. Now it contains only protein molecule, so we just have to head over here, click save PDB and navigate to the desired folder and then save it as a clean protein. This will be input for ScientiFlow to perform docking. You have to upload a CSV file that will contain list of proteins against which list of ligands to dock. This CSV file will serve as a configuration for performing docking. Content inside the CSV file will change according to the type of docking. Let us download them to explore one by one what you have to fill in each cases. For blind docking, provide the list of file names of proteins against which file names of ligands to dock and upload these protein and ligand files in these folders respectively. For site-specific docking, in addition to the protein and ligand information, you also need to provide either the co-crystallized ligand information or the grid parameters for the site-specific docking. To prepare the reference ligand, let's say through literature, you know there is a drug molecule co-crystallized with the target protein available in PDB. Download it, open it with Chimera like this or Pymal. Let's say my drug molecule is 4L5. Go to select, look for residues and you will find the drug name 4L5. Select that one, invert all the selection. You will be able to select all other molecules except your drug molecule and delete everything. Now if you save this file, you will be able to extract only the reference ligand and then save it properly. This reference ligand has to be uploaded inside this reference ligands folder. That file name has to be updated here properly. On the other hand, if you have generated grid parameter file through AutoDock, you will get a file something like this. Please remember, it will be beyond the scope of this tutorial to cover the grid parameter generation through AutoDock from known residues. These three values placed here in the grid parameter file, that is corresponding to the center, has to be placed here as center x, center y, center z. These three values present here NPTS has to be multiplied with the spacing and then placed here as size x, size y, size z. So each of these NPTS values have been multiplied by the 0.375 and those values are reported here. That's it. These configurations are sufficient for performing site-specific docking. Now I will show you demonstration to perform site-specific docking, since blind docking is very simple. Let's get started with practicals. Let's say we have 5 proteins and 12 ligands and want to perform cross-docking. It would have taken 60 docking for us to complete our study. Performing docking and protein-ligand interaction manually would have been difficult. Let us see how simplified it is in ScientiFlow. I have cleaned and placed the protein files like this here. Cleaning was done as I described earlier. I have placed the ligand files here. For 3 of the proteins, I have co-crystallized ligand which are placed here. And these information have been typed here. For 2 of the protein, I had to generate the grid parameters manually via AutoDock, and so I placed those values here. To perform cross-docking between 5 proteins and 12 ligands, let us open a fresh CSV file. In this one, copy and paste the headers from the previous one. So I am just copying the headers and pasting it here. Now copy and paste the first protein details. Now copy and paste the ligand details. I am pasting all the 12 ligand details. Now just replicate this one. Now copy the next protein detail. Now copy and paste all the ligand details as well. Now replicate the same process for all the proteins and ligands. Now I repeated the process for all the proteins and ligands and got the totally 61 records, that is including the one header. So totally this will perform the 60 docking, that is between 5 protein and 12 ligands to make 60 cross dockings. Now I will configure the workflow for performing docking. Since I am going to perform site specific docking, I will toggle this on instead of this one. This is to be toggled only if you are performing blind docking. And here for the scoring function, I am going to choose Vina scoring function. And then rest of them seems to be fine. I am just going to save this configuration. Now it's time to upload our samples to the ScientiFlow. First let's upload the proteins file. You can delete this existing file or leave it as it is. I am going to upload as it is. I am going to upload all my protein files. And then all my ligand files. This will upload 12 ligands now. Reference files. This is the co-crystallized ligands that I am going to upload now. Now finally, I need to upload the CSV file. ScientiFlow cross-docking is the CSV file I created as the name. That's it. Now we have uploaded all our samples. We just have to head over to the job section. We'll create a new job. In this I am going to say that cross-docking as the job title. Here I am going to write the job directory inside the projects folder, this job directory name will be created. So keep the job directory without empty spaces. Keeping without empty space is good practice in the Ubuntu environment. Now choose the start node of the workflow. I am going to choose from the start of the workflow and then end node of the workflow. You have to choose till the end. For this docking example, I am going to perform site specific, so it is already toggled in, and I am going to choose Vina scoring, so it is already toggled in. This is just an environmental variable which is dynamic variables. So I am going to choose the exhaustiveness and num modes here, which is by default it is 8 and 9. I am going to leave it as it is. For the input variable, I am going to give ScientiFlow _ cross _ docking.csv file. This is the file we have prepared as a sample to be performed docking. So mark this one and then select this file. Now just click continue and then submit the job. I hope everyone has completed the 3-step process during the start of this application. If you have completed this one, now head over to the terminal and you will be knowing how to start the ScientiFlow jobs. To list the pending jobs, first thing you can do is ScientiFlow CLI flag help. This will list all the commands that are possible within the ScientiFlow. To list the pending jobs, just type the ScientiFlow CLI and flag list jobs. This lists the pending job that we have recently submitted, that is 540 job ID, that is cross docking that we submitted now. To execute this particular job by ID, just type this particular command, that is execute job by ID. ScientiFlow CLI flag execute job by ID 540. If you trigger this, it will start pulling all the relevant files and also the containers that are required to run this workflow. Since the containers have been already downloaded, it is just using the cached images of these containers. But if it's downloading for the first time, make sure that your internet is stable to download these containers. Now this is performing site specific docking for all the complexes that we are uploaded. Wait patiently, since it is going to perform 60 docking, wait until its completion. Now the docking workflow has been completed successfully. I hope during the getting started process, you would have configured the base directory somewhere. I did it here. In this folder, you will find your project title. Mine is demo docking, so I am just navigating inside this. Inside this folder, you will see the project job folder that we created, that is cross docking that we created. Inside this, we will have all our project files like protein files, ligand files, output files, reference files and everything. Now navigate to the output folder. You will see a CSV report at the end, that is docking report.csv. If you open this file, you will see all the docking scoring with the binding affinity for each of the combination and also the number of bonded interactions between them. This report is available for all the 60 complexes. And each of these reports, each of the docked complexes are available inside these subfolders. You can simply navigate and then open the files. You will see the all confirmation, best confirmation, dock complex and GNINA docking report. If you open dock complex, you will see the docked complex between the protein and ligand for each of them. Likewise, you will have all the individual complexes for each of these cases properly organized. That's it. Hope you all learned to perform several methods of docking with varied scoring function under one roof in ScientiFlow. Thank you.